right. In the interest of being on time, I'll go ahead and get started. Everybody will get all settled in. So on behalf of Dr. Marie Bleakley, a co-moderator, as well as our very, very esteemed panel, we'd like to thank Dr. Wade Kiono and all the Siop organizers for inviting us from Seattle to present to you an educational session on novel therapeutic approaches to high-risk B-cell ALL. We're so fortunate to have on our panel national leaders in the setting of therapeutic principles of B-ALL, biology and discovery in B-ALL, and in cellular immunotherapy and stem cell transplantation. We look forward to sharing insights from our entire team in this educational session. So here are the presentations. First, Dr. Lowe will talk to us about optimizing risk stratification through new genomic discoveries. Dr. Lowe is currently the division head of hematology, oncology, bone marrow transplant, and cellular therapies at Seattle Children's and directs our Bentown Center for Childhood Cancer and Blood Disorders Research. As many of you in this room know, her research focuses on the genetics and risk stratification of both ALL and NJMML. She's a past chair of the Children's Oncology Group ALL Committee and supervised the clinical trial portfolio within COG for newly diagnosed and relapsed ALL. Next, we'll hear from Dr. Rachel Rao on optimizing treatment approaches to maximize cure and minimize toxicity. Dr. Rao is an associate professor of pediatrics at the University of Washington. Her lab-based research is focused on understanding the molecular mechanisms that drive leukemogenesis and therapy resistance and translating these findings into clinical applications. Within the Children's Oncology Group, she has served as a chair or a vice chair of several studies, including the current standard risk B-ALL trial, AALL 1731, and she also serves as the COG ALL Vice Chair for Biology. Next, we'll hear from Dr. Colleen Annesley about recent advances in cellular immunotherapies in B-cell ALL. Dr. Annesley is an associate professor of pediatrics at University of Washington and leads our cellular immunotherapy efforts at Seattle Children's and the Seattle Children's Research Institute. And she works on translating these therapies into the next generation of clinical trials. And finally, we'll hear from the co-chair of the session, Dr. Marie Bleakley, on recent advances in stem cell transplantation and B-cell ALL. Dr. Bleakley is a professor of pediatrics at the University of Washington and a transplantation and cellular therapy physician scientist at Seattle Children's and the Fred Hutchinson Cancer Center. She co-directs our high-risk leukemia program at Seattle Children's, which provides personalized and integrated treatment plans and research studies for children with high-risk leukemia. So these are our educational objectives, which are really embedded into the titles of each of their presentation. And I can leave this up here until the next talk. So first we'll hear from Mignon.
They'll bring it up? Okay. We'll wait till that comes up. Thanks, Todd, for that great introduction. I want to thank you and Marie for inviting us. And I also want to thank Wade and the organizers of the conference. It's wild just to watch this. Okay, great. All right, so it sounds like it's going to be a hit parade on ALL. Let me just this afternoon. So I'm just going to start us with a discussion on how we can optimize risk stratification through some of the new genomic discoveries that actually have been very recently published in the Journal of Clinical Oncology. This is my disclosure slide. And I always start the presentations that I give on ALL with a slide that I'm particularly proud of. This looks at survival over time in years from diagnosis over different eras of clinical trials that we've had our patients participate on. And these curves are no different from curves from the Dutch group, the BFM group, the St. Jude group, the Dana-Farber group. We've done a remarkable job, I think, over the last 50 years in really achieving phenomenal cure rates for children with ALL. These are the outcomes of children specifically enrolled on Children's Oncology Group trials, Legacy CCG or Legacy POG trials. And we're really getting to where we are achieving substantial rates of cure for children who are newly diagnosed, and we have very positive conversations with our patients. How have we achieved these cure rates? We won't really speak today about how we have delivered multiple chemotherapy agents to prevent drug resistance, nor will we talk about how we had to recognize that sanctuary sites needed focused treatment like the CNS. We did, I think, a lot of work over the last 20 to 25 years on identifying risk groups to determine the intensity of therapy, such as the NCI risk group. We've identified sentinel genetic lesions that predict outcome, and we've also incorporated early response to therapy in how we determine how intensely patients are treated after their first month of induction. Cures have evolved then with risk stratification and chemotherapy intensification. And modern risk stratification really categorizes patients into post-induction groups to receive treatments balancing perceived relapse risks with the intensity of therapy. And the risk categories used by the Children's Oncology Group include age between 1 and 9 years or greater than 10 years, a presenting white blood cell count less than or greater than 50,000, leukemia genetics, which is bolded because that's what the topic of my talk will be today, minimal residual disease measurements, and at least in the COG, we use flow cytometry in peripheral blood on day 8, and we also use it in the bone marrow on day 29, and CNS status. And the question is really, how can we improve on current risk stratification? So I'm going to review some of the work that we recently generated from a molecular profiling to predict responses to therapy project that was part of then Vice President, now President Biden's Moonshot initiative to really tackle cancer. And for those of you who are not familiar with President Biden's family history, his son tragically died from a brain tumor a number of years ago. And as part of that grieving work, I believe, he actually really wanted to focus federal funds on identifying ways that we could advance knowledge in cancer. And we were awarded a contract to do a project, and the title of that project is Genomic Determinants of Outcome in Acute Lymphoblastic Leukemia, and it was a Children's Oncology Group study. It was based on the fact that when we run our clinical trials in COG, we also bank a number of samples from both diagnosis and the end of induction when most patients are in remission. And we have all of those samples that are closely tied to patients who participate in our clinical trials. So we have outcome measurements, and we also have potentially some measurements by disease, arm, et cetera. So the background of the study, the reason why we did this is that we knew at the time, and this started in 2019, 2020, that patients with NCI standard risk B-ALL, so those are the children who are between the ages of 1 and 9 and present with a lower white count less than 50,000, they actually, when they do relapse, they account for approximately half of our relapsed cases. And we also know from a lot of the work that we have done in the past that genomic predictors of relapse in standard risk B-ALL patients had not been comprehensively studied in large numbers. A lot of the work that we had published in collaboration with WashU, PCGP, and St. Jude, et cetera, had focused on high-risk subsets of patients. And because this is such a burden for patients with relapse, we wanted to see if there was any way that we could actually predict who might actually relapse when they presented with standard risk features. We also knew that up to maybe 20% or so of patients enrolled on one of our comprehensive relapsed ALL trials, AALL 1331, actually presented with ETV6-RUNX1 or double trisomies of chromosome 4 and 10 at relapse. And so we really wanted to know what made these particular patients different as well. So we had a limited budget, actually. We could analyze up to 1,500 patients, and so we identified 1,496 who were enrolled on a number of standard risk and high-risk ALL trials. We did deliberately try to focus on standard risk patients, so that was almost 1,400 patients. And then we also had a smaller subset of high-risk patients who we knew had favorable genetics, which we count as those patients with double trisomy of chromosomes 4 and 10 or ETV6 RUNX1. We actually also deliberately designed it as a case control design, which proved to be challenging when we were now analyzing the data. And the reason is because most of our standard risk patients do quite well. We really wanted to make sure we were identifying factors that were enriched in those patients who relapsed. So a case was a patient who relapsed, and the control were patients who remained in continuous complete remission for at least 5 years of follow-up or at the last follow-up. They were also matched on MRD at the end of induction. And I would just say that it was in a 1-to-2 design, so it was one case to every 2 controls. And when we did the analysis, we had to wait for the case control selection. So we were able to do genomic analysis based on sequencing data using whole genome sequencing, whole exome and transcriptome sequencing, and we characterized the associations between lesions and status of relapse in MRD. And so you see the platforms on the left, looking at the somatic lesions, which is what we have mostly focused on so far, and then we analyzed what was different between those patients who ultimately relapsed, so we didn't actually sequence their relapse specimens, but what was different at diagnosis between those patients who ultimately relapsed and those patients who stayed in remission. And again, these analyses were weighted for the case control selection. The most prevalent subtypes, which is not unexpected, were those patients with ETV6 RUNX1 or hyperdiploid ALL. This is just a t-SNE plot using a previous algorithm St. Jude had optimized, and you can see the samples here showing that they segregated very nicely with their genetic fusions. And we also, and this is a bit small and I'm sorry, looked at all of the driver lesions. The red bars indicate those numbers of patients who relapsed, and the gray bars indicate those patients who remained in a continuous remission at the time of publication or the time of last follow-up. The ones on the left, the numbers are much bigger because they were the more common lesions, and the ones on the right are just the less common lesions. But again, just important to remind you, this is not a generalized retrospective whole cohort. This is a case control study. We then looked at clinical factors and genetic subtypes associated with relapsed and MRD status. And as you can see, so in this plot, the numbers that are in red show a significant association. Sample sizes greater than 10 are actually shown because, again, this paper is rather dense. But what you can see, for example, is that there's a negative association between your presenting age between 3 and 5 for relapse and your day 29 MRD. And there's increased relapse in the following categories. If you look on the sort of left panel, hyperdiploidy without double trisomy, a PAX5 alt subset, which we'll speak about in just a moment, ETV6 RUNX1-like, so they do not have ETV6 RUNX1, but they have a gene expression profile that's not that far off, and PH-like. I'm just going to talk about 3 particular genomic examples that we're thinking about incorporating into our next generation of trials, and we'll talk just briefly about that at the very end. We did identify that PAX5 genomic alterations are associated with increased relapse in standard risk ALL. It's really the first observation that up to even 49 or 50% of PAX5 alt patients who present with standard risk ALL actually experience relapse. The panel on the left shows those patients and their genomic lesions in PAX5 that relapsed, and the one on the right shows those patients who did not relapse. We also found that certain novel PAX5 alterations did occur in standard risk ALL, such as intragenic deletions and amplification of PAX5, as well as PAX5 null 4L fusions. We did determine, and our numbers are getting a little bit smaller now, that PAX5 amplification is associated with an increased risk of relapse in the PAX5 altered subtype, and that the focal internal tandem PAX5 amplifications always involved PAX5 exon 5, and that these tended to be associated with the worst outcomes. And I say tend because, again, the p-values were a little bit less than optimal just because of the numbers. And that we also know, again, when you look at the PAX5 amplification, it's associated with increased risk of relapse in the other PAX5 altered subtypes. So focal internal, as I said, PAX5 amplifications always involved exon 5, and again, PAX5 amplification was associated with the worst outcomes. So the second example I just wanted to present, and I'm sorry, we only have about 15 minutes to talk about these data, but was ETV6, RUNX1. So we know that's one of the most common fusion genes we see in childhood ALL. It generally has outstanding cure rates, both disease-free survival and overall survival, but as many of us know, relapse does occur. This was a large effort that is going to be published soon, and actually I think it is in press. Sue Reingold and Deepa Bhojwani led a study where they actually looked at outcomes after relapse in a number of patients, 25,000, with newly diagnosed ALL. And you can see the split between those who did not have ETV6 RUNX1 and those who did. We actually looked at these breakdowns in patients who were enrolled on that umbrella relapse trial, AALL 1331, and found that 13.5% of the patients actually had ETV6 RUNX1. And so the question was whether we could identify which ETV6 RUNX1 positive patients at diagnosis had a higher risk of relapse. And we found a few sort of interesting things. Basically, we identified that the risk of relapse in ETV6 RUNX1 is higher than in those with unbalanced translocations. And you can see the probability of event-free survival on the right. We also found that unbalanced ETV6 RUNX1 translocations with a gain of RUNX1 or ETV6 RUNX1, so double the fusion, were more common in standard risk patients who relapsed. So you see the balanced versus unbalanced, which was the same as the last slide, and then the derivative 21T1221. We then wanted to ask, well, are there other secondary alterations within the ETV6 RUNX1 positive patients that might predict relapse? And so we started to look at other potential genetic abnormalities, not only across the entire cohort of patients who relapsed, but also in patients with distinct driver genetic subtypes. And we found that alterations of INO80 were associated with relapse in ETV6 RUNX1 positive standard risk B ALL. These were significantly enriched in patients who relapsed. And we also, we're continuing to do some studies, but we know that this particular gene, which can be associated with chromatin remodeling, is also physically close to MGA on chromosome 15. When we looked at the entire cohort to see what were the most common predictors of relapse, we see in panel A what might have an increased risk of relapse, and then we looked at distinct genetic subtypes, which is coded in those coded bars in panel B. And again, we find that for patients who had ETV6 RUNX1 positive disease, the presence of an INO80 alteration was associated with an increased risk of relapse. The third example that I just wanted to present is that we, of course, validated that mutations in the IKZF1 C-terminal zinc finger was enriched in the relapse cases. So mutations in the C-terminal zinc fingers have were observed almost exclusively in those who relapse. And then samples who had the dominant negative phenotype have the worst event-free survival than patients with other types of deletion or no deletion. And again, you can see the red bars are those patients who relapsed, and those in the gray bars are those patients who remained in remission. We also looked at the presence of IKZF1+. So remember, these are patients that largely had standard risk ALL. Martin Stanula's paper in 2018 defined what IKZF+ was, and it is the deletion of IKZF1 with an additional deletion of essentially CDKN2A, homozygous CDKN2B, PAX5 or PAR1 with retention of ERG. And so we wanted to look at the same sort of constellation of genetic alterations to determine if, again, this was also associated with relapse in our standard risk patients. And so samples with the IKZF1+, basically defined by the MLPA definition that many institutions in Europe use, had the worst EFS amongst patients with IKZF1 alterations than patients with other types of IKZF deletion or no deletion. So how can we use this information to improve on current risk allocation for future trials? Well, one thing that the committee is now discussing, and I can observe from the side, which is great, is that only the best performers who can be in the most favorable risk or the lowest risk group, these patients might be getting, reduced therapy. We've come a long way in curing these patients, and now we're thinking that it may be time, as many other groups are considering, who can actually get slightly less intensive therapy and still maintain their same rate of disease-free survival. And also importantly, I'm going to go off script here for just a moment, their overall survivals are really excellent too. And maybe that is something we really should be thinking about as we move to future trials, is to focus on overall survival. So for example, the ETV6-RUNX1 patients with the unbalanced translocation or an INO80 lesion, maybe they're not the ones to be in the most favorable risk group. Maybe they need to receive some more intensive therapy. And then others might need more intensive therapies, more than just standard risk average. Maybe they get upstaged to receive high-risk backbone therapy, like those who have the presence of IKZF1+, regardless of their MRD, or the dominant negative IKZF1 lesion, and those who have PAX5 alt disease. And of course, all of this actually depends on the therapeutic question, and it may get very complicated with the emerging data on blinatumomab. But I think in general, our goal is to really identify what phases of traditional cytotoxic chemotherapy can we safely remove and still maintain our outstanding outcomes. So in conclusion, I feel that the MP2 PRT study does provide new information on the risk of relapse in NCI standard risk patients and some NCI high-risk patients interrogated with comprehensive genomic approaches. We identified some new driver lesions that were found to be associated with a higher risk of relapse. We also know that secondary lesions influence this risk. And we believe that these new lesions can be incorporated into new risk stratification algorithms that will continue to reduce therapies for patients with the most favorable features and intensify or change therapy or modify therapy for those with less favorable features. And the plans currently, which you may hear about more from Dr. Rao, is to really perform comprehensive sequencing that will allow us to move the needle closer to curing every child with ALL by tailoring our intensity of therapies. So I just want to acknowledge this is not a sufficient acknowledgement. It really has taken a village to run the ALL trials, so it's just a small number of people who've been involved. And obviously we are incredibly indebted to our patients and their families for allowing us to enroll them on our clinical trials. And this is just the MP2 PRT team. So thank you very much for your attention. And I don't know if I can take questions or we can wait till the end. So. Oops.
But we have time for probably one question, and then we'll have questions afterwards.
Sorry, I don't mean to show you the characteristics of... Okay. Hello, Peter.
In this study, did you look also at germline variants?
That's a great question, and we're just getting the energy up to start doing that. It's a very interesting question, and we would like to do that. It's important, yeah.
Okay.
Great, thank you.
Thank you.
All right. Thank you, everyone, for attending, and thanks, Todd and Marie, for the invitation to speak today, and Wade for organizing the conference and allowing this session to happen. So my topic of choice was optimizing treatment approaches to maximizing cure and minimizing toxicity in B-ALL, which is a pretty broad topic, so I'm going to try to stay focused on a couple of ways in which I think we can achieve that ultimately moving forward. Those are just my disclosures. So this is a slide that Mignon just showed, and I think many pediatric oncologists love to show this slide because I think it shows that B-ALL is really a paradigm of medical research success over the decades, with these large increases in outcomes achieved for our patients over the last several decades. But I think I would submit that over the last couple of decades, so since the early 2000s, we've sort of reached a plateau in the outcome improvements that we've been achieving with each successive iteration of trial. And certainly that's not for a lack of effort or trying. The COG and other consortia around the world have tried strategies to continue to push these outcomes even further. But at least the last iteration of COG studies that we ran, one thing that we learned is that further intensification of standard chemotherapy is not the way forward. We see that we incur an excess of toxicity without a return in efficacy that makes that strategy not a feasible one to move forward from here. And so I think as we've designed the current iterations of COG studies and now the subsequent ones to come, we've kept this paradigm in mind that we really need to have novel approaches to really be able to move the needle forward from here. So how do we do that? I think Mignon just gave a great talk about how we can use novel genomics to more precisely risk stratify our patient, matching the patient's risk based on clinical features and genomic information to better attune that to what chemotherapy backbone they might need for optimal cure and to lessen the amount of toxicity. So those who are readily cured with relatively low intensity chemotherapy, it's as important to identify those patients as it is to identify patients at higher risk who need a greater intensity of chemotherapy. So I won't focus my time here on that, but two other sort of broad categories of ways I think we can move forward include targeted approaches for certain high-risk subsets, as well as leveraging the immune system. And so I'll talk about those briefly today. Okay, so I think we've all sort of reaped the benefits of what imatinib and dasatinib and other TKIs have done for pH-positive disease. As you know, this is driven by the BCR-ABL kinase, and it was discovered in the late 90s to early 2000s that you could have small molecules that bind in that pocket to prevent that activated signaling kinase that leads to the downstream gene expression program that fuels pH-positive disease. And the addition of TKIs revolutionized the outcome for these patients from a largely incurable disease to one in which we're achieving now disease-free survival in excess of 70% and overall survivals in excess of 90%. And this was the initial COG study that demonstrated continuous TKI plus chemotherapy improves outcomes substantially. And the subsequent two studies done showed very similar results, independent of which TKI you use and which chemotherapy backbone. But I think one of the things to take note is that we added TKI to rather intense chemotherapy backbones. Most of us consider sort of what we call the SFAL backbone to be a standard of care for such patients. And I think that really came from a time in which we really had to maximize potential outcomes for such patients with just chemotherapy alone. And the logical next step in studying that was to add TKI to that existing standard of care backbone. The problem with that, of course, is that continuous TKI plus this chemotherapy backbone is associated with, excess mortality. There's about 5% of patients that will have remission deaths secondary to mostly infectious toxicity. So the most recent study of this, run in collaboration with the Children's Oncology Group and European partners, was AALL 1631, chaired by Louis Silverman out of the Dana-Farber. And this asked if we could maintain those really favorable outcomes of pH-positive disease, yet lessen the chemotherapy intensity to reduce that excess toxicity that we're seeing in these patients. And so this is a randomized trial comparing our standard of care SFAL backbone plus continuous TKI to a slightly more gentle modified BFM chemotherapy backbone. So unfortunately, I don't have results to share from this yet because this trial just closed earlier this month. And so hopefully we'll learn more. And I think we're all very hopeful that giving less chemotherapy plus continuous TKI will result in similar excellent outcomes that we've become accustomed to in the last several years. Now, of course, pH-positive disease has a counterpart. We know based on really elegant gene expression program profiling of multitudes of BALL cases from both the US side and European and other consortia, that there's a class of BALL that has a gene expression program that's essentially overlapping with pH-positive disease but lacks the BCR-ABL fusion. And those are largely driven by fusions of other kinases, both ABL kinases and JAK-activating kinases. And so we also know that like pH-positive disease, with standard chemotherapy alone, such patients tend to have suboptimal outcomes. And so a large question, not only by the Children's Oncology Group, but other consortia has been, can we improve outcomes in these patients similar to what we've done with pH-positive through the addition of targeted kinase inhibitors? And so that would be imatinib or dasatinib for our ABL class fusions. And that was studied first on AALL 1131 in the COG. There was a dasatinib arm towards the end of that trial. And then the 1631 trial that I just described for pH-positive patients included patients with Abel class fusions. Additionally, the trial that's currently being redesigned, I should say, but hopefully will open within the next year or so, is ALL 2131 that will also be studying the impact of TKI on the outcome of such patients. Additionally, while imatinib and dasatinib can target the Abel kinases, ruxolitinib, a JAK inhibitor, can potentially target the JAK-activating pH-like disease, and that was studied in the COG trial AALL 1521, which closed to accrual last year. So unfortunately, I don't have any data to share, but we're very hopeful that with the addition of targeted interventions, such patients can achieve improvements in outcomes similar to what we've seen for our pH-positive diseases. So much more to come, hopefully, very soon on these patients. Okay, another group of patients that we know have suboptimal outcomes with our standard chemotherapy alone are those with KMT2A rearrangements, formerly known as MLL. This is obviously a subset that's incredibly common amongst our infants with BALL. Over 80% of those patients will have KMT2A rearrangements. And then while less common amongst our non-infant BALLs, it's associated with an inferior outcome amongst those patients as well. So over the last 10 years or so, there's been really exciting, I think, advancements in the field. The MLL is an epigenetic regulator, and so a lot of efforts have gone into trying to target the epigenetic program that is driven by the MLL fusion genes. And so perhaps the most successful of those has been targeted inhibitors of the MLL interaction with another regulator protein called menin. And for the MLL fusion to function and to sit down and turn on gene expression that leads to the BALL phenotype, it has to interact with menin, otherwise it's ineffective. And so a number of pharmaceutical companies develop small molecule inhibitors that sort of sit in that menin-MLL interaction pocket and prevent them from functioning together, and so therefore shut down that gene expression program that's driven by the fusion proteins. And so I think rightly so, the menin inhibitors have led to quite a lot of excitement, and there's a lot of emerging data from early phase clinical trials. There are a number of these agents being developed, and they're in various staging of the clinical pipeline. The furthest along is Syndex 5613, or revumenib. So just very briefly to cover some of the early phase data that we have from revumenib and sort of why it sets the stage for what I think will come next. The initial trial, Augment 101, was done in pediatric and adult patients with relapsed refractory KMT2A rearranged leukemias. This could be AML or ALL. And just by nature of sort of this patient population, this was enriched for AML patients, but did include ALL patients as well. And the phase 2 portion of this trial only enrolled patients with KMT2A rearrangements who were all, varying ages, but median age in the young adult group. So it did include a fair number of pediatric patients. And these are all very heavily pretreated patients, many of whom had already undergone a first stem cell transplant. There are almost 60 that were available for efficacy analysis. And I think quite strikingly for monotherapy of a heavily pretreated population of patients, the CR rates were quite impressive, more so than we're used to seeing with monotherapy early phase trials. So this is, I think a really promising efficacy signal that warrants its incorporation into the therapy of KMT2A rearranged patients. There are some unique toxicities to these, but overall they're quite well tolerated. Some GI toxicities are the most common. QTc prolongation is also seen amongst patients treated, about a quarter of whom will have grade 3 or greater. And then for AMLs in particular, this can result in a differentiation syndrome. So on the backbone of those impressive monotherapy results, a number of trials are now studying revumenib or other menin inhibitors in combination with chemotherapy. The first one there, Augment 102, so the next step from that Augment series of trials, showed pretty impressive results in terms of CR rates, most of whom were MRD negative, with a combination of FLA plus revumenib, and then a number of other trials are coming down the pike showing really impressive efficacy data. So I think these are really promising agents. To the point where we've now opened an infant KMT2A rearranged BALL trial, AALL 2121. This is open and enrolling. It's studying 3-drug induction or FLA in combination with revumenib for infants with relapsed KMT2A rearranged leukemia. So really exciting to have this agent available to this very difficult to treat subset of patients. Additionally, we've got a very, I think, exciting collaboration with the SWOG group. It's going to be an intergroup trial that will include COG and SWOG that will actually look at revumenib in the upfront setting. So all the studies that I've talked about so far are in the relapse setting, but I think with the promising results from the relapse setting, it makes sense to see if we can improve outcomes when putting it up front. Okay, so with the last part of my talk, I'm going to focus on how we can leverage the immune system. And I won't get a chance to talk about sort of all the exciting things, but I think we've learned over the last decade to 15 years that while chemotherapy can get us so much, I think we've got some exciting options to think about in how we can leverage the patient's own immune system. So, of course, blinatumomab is a bispecific T-cell engager. One component of it binds to CD19 on the patient's lymphoblast, and the other component of it binds to CD3, so engages the patient's T-cells for cytotoxic killing of the B lymphoblast. Inotuzumab is an antibody-drug conjugate, so the antibody binds to CD22 on the surface of B cells, and then it has a cytotoxic payload, colchicamycin. So when the 22 antibody binds to the cell surface, it's internalized, and the colchicamycin is released, activated, binds to the DNA, and leads to cell destruction. In the interest of time, I'm not going to talk about the data for inotuzumab today, but suffice it to say, it's shown great promise in the relapse setting. It has a very overall good tolerability profile, and so it's also now being studied in the upfront setting. And then lastly, of course, cellular therapies, which Dr. Annesley will talk about in much more detail, but CD19 CAR T cells have shown great promise in the relapse setting and are now making their way into the upfront setting as well. All right, so the last part of my talk, I'm going to focus on the data to support blinatumomab for the treatment of BALL patients, and we'll start with the relapse setting. So initially, there was a phase 1/2 trial led by the COG in collaboration with European colleagues, looking at single-agent blinatumomab in the relapse refractory setting, showed a really strong efficacy signal and a very favorable safety profile, and so very quickly moved into our relapse population in a study AALL 1331, which was for first relapse of BALL. At the same time, a trial was being run in Europe, which was sort of a very similar approach. And for our high-risk relapse patients, 1331, after an R3 reinduction, gave patients either 2 blocks of traditional intense chemotherapy or 2 blocks of blinatumomab followed by a stem cell transplant. Similarly, the European study that was run at the same time, after reinduction, patients got 2 cycles of intense chemotherapy, followed by either 1 more cycle of intense chemotherapy and transplant, or blinatumomab followed by transplant. And quite strikingly, both these studies came to the same conclusion. Blinatumomab was far better than standard chemotherapy for the populations of patients being treated here, both in terms of disease-free or event-free survival, shown there, and overall survival. So more patients were getting to transplant, more patients were achieving ultimately a cure when treated with blinatumomab in replacement for those intense chemotherapy blocks. Also, probably not surprisingly, blinatumomab was far better tolerated compared to those intense chemotherapy blocks with significant and really, I think, striking reductions in particularly infectious toxicity in patients who got blinatumomab as opposed to those who got standard chemotherapy. Additionally, the 1331 trial also included a low-risk relapse arm, so patients who were late relapses and had a good response to initial chemotherapy were allocated to this arm, where they are randomized to receive either block 2 chemo followed by block 3 chemo, which is also very intense, 2 continuation blocks of maintenance, or sort of standard of care, versus that minus the block 3 chemo with 3 cycles of intercalated blinatumomab. These results were very recently published by Laura Hogan, and it showed a trend towards improved survival for the whole population treated on this low-risk backbone when given blinatumomab. But if we look specifically at those with bone marrow relapses, they had a significant improvement in survival. And this is likely accounted for by the fact that we know blinatumomab doesn't have great CNS activity, and so patients who were isolated CNS relapses did not benefit from blinatumomab, so when you take them out, I think that's where you really see the effect of blinatumomab. Okay, so I think this leads to the next set of trials, and I think what we're all excited about is now we've taken away some of the bits of chemo there, but can we take away more? And there's a really exciting data that was generated by the UCAL group And the question they ask is, can we sort of start with this sort of gentle reinduction approach where we're more likely to debulk disease, leading to transplant? So can we be more gentle with our reinduction therapy? R3 is notoriously quite intense and associated with high rates of toxicity, so can we do better there? So this was designed in that younger patients got 3 drug induction and older patients got 4, and then there was a check of MRD at day 15. For those who are already less than 5% MRD at that time point, they could stop induction therapy and go on to get blinatumomab, and that was about 50% of the patients. The remainder completed the subsequent 2 weeks of induction, and then they proceeded to get 2 cycles of blinatumomab. High-risk patients went on to transplant. Center-risk patients went on to get a DFCI-style delayed intensification, followed by just maintenance therapy for a total of 2 years. And while the follow-up time is relatively early, I think what you'll note is that the results are actually quite good. So for those high-risk patients who got pretty minimal reinduction therapy, blina, and transplant, their outcomes are pretty comparable to what were achieved on 1331. And then the standard-risk patients who, again, just got 2 cycles of blina, followed by pretty gentle, by comparison, chemotherapy. Their early outcomes are quite good, event-free survivals of 84, and overall survival of 100%. So I think this opens the door for potentially further reductions in relapse therapy. Okay, so how about in the upfront setting? We've talked about the efficacy and tolerability of blina in the relapse setting, which sets the stage to move it into the upfront setting, of course. So I think one of the first trials that caught everyone's attention was the use of blinatumomab for our infant B-ALL population, and this was a study led by Inge van der Sluis. And what was done is that patients got the interfog chemo backbone plus 1 cycle of blinatumomab, and the results were the likes of which I think we'd never seen for infants with B-ALL. You'll note that their event-free survival is in the 85% range compared to historical control treated with the same chemotherapy backbone minus blinatumomab. Additionally, more recently, the adults have studied the addition of blinatumomab to a pediatric-inspired chemotherapy backbone and seen, really striking improvements in outcome. And then in the Children's Oncology Group, we recently conducted 1731, which randomized patients to receive their standard chemotherapy backbone plus or minus blinatumomab. Unfortunately, I can't discuss those results. The data are under embargo, but hopefully more to come soon on that. All right, so in the last little bit here, in the relapse setting, we've been able to take away some of the chemotherapy and replace it with blina. We haven't yet tried that in the upfront setting in a randomized clinical control trial. We've added blinatumomab to standard chemotherapy and seen good outcomes, but can we now take away some chemotherapy and replace it with blina? And I think there are at least 2 examples that show us that's probably very likely. So a French study looking at pH-positive adult patients took a very interesting approach. They gave patients a little bit of steroid, followed by dexamethasone and dasatinib, followed by 2 to 4 cycles of blinatumomab plus dasatinib. So almost really no standard chemotherapy, steroid, TKI, and blina. Now, as you might know, adults consider most patients with pH-positive disease eligible for transplant, so that was allowed in this trial, and a pretty big number of patients were transplanted. But the outcomes were quite good, in excess of 75% disease-free survival, which is excellent for an adult population, much less one who gets such little amount of chemotherapy. Also important to note is that they do report on the outcomes of 28 patients who just got this and were not transplanted. They didn't get any further chemo, and 27 of those patients are still in complete remission now 4 years out from therapy. So I think incredibly promising that we can really cure some patients with minimal standard chemotherapy with targeted approaches and immunotherapy. And then lastly, some data out of UK. This wasn't a clinical trial, but they developed clinical guidelines. I think we've all had patients who you just can't give them that next chemotherapy because they're too sick, they've got too much toxicity, they have Down syndrome or some other disorder that leads to believe they're not going to tolerate chemotherapy, and so we give them a blina bridge to get them through, to help them heal, to give them time. And they looked at this in a systematic way. So they looked at patients who completed either all of induction and part of consolidation, or all of consolidation, but then couldn't proceed on to the next planned chemotherapy, and gave them 2 cycles of blina instead, 1 to 2 cycles of blina. Some of them went on to transplant if they met eligibility criteria for that. Others got DI and maintenance, and some just got maintenance after those 2 cycles of blina. Those of the 20 who were transplanted, 15 still remain in continuous complete remission. Some died of infectious toxicity, some relapsed. If we look at those who got DI followed by maintenance, all of those patients are still in remission, and that was 64. And then those patients, to me, that are most interesting, the ones that got some induction, blina, and then maintenance, of the 18 of those, 17 remain in complete remission. And the one patient who relapsed literally got 2 weeks of pred as his induction before he got blina and then maintenance. So, I mean, it's probably not surprising that that child relapsed. But I think this is really encouraging to say, yes, we can replace some, if not a lot, of chemotherapy with blinatumomab for upfront populations. And I think that's where we're moving as a field. All right, so to wrap it up, with targeted agents, I think we have opportunities to improve outcomes for historically poor prognosis groups of patients. Certainly pH positive, the jury's still out for pH-like. And then KMT2A, I think we finally got something that's tractable that can improve their outcomes. Ultimately, I think targeted interventions can allow us to dial back some of the chemotherapy as well. And then with immunotherapy, we've shown that this is highly effective in relapse and is now part of the standard of care there. And the evidence really mounting for its use in the upfront setting, with the future directions of really taking away many of the elements of toxic chemotherapy and replacing with immunotherapy. And so this is my future directions. I think we don't want to stop there. We've got room and we want to hit that. And I really think that we're looking at a realistic future date to achieve that with the combination of approaches that we're all talking about. So I'll stop there and be happy to take any questions.
We have time for one question, and then again we'll hopefully have time at the end to ask more.
Hi, just one question. So first, the data were very impressive and compelling. This is Vic from India. For LMIC countries, in terms of what is the cost comparison for blinatumomab? Because we find it unaffordable, literally. It's like about, I think 4 years or 5 years worth of salary. So it's a challenge. So yes, within high-income countries, definitely it's having a huge impact, but it's not translatable to the majority of countries around the world.
Yeah, I would fully support that statement for sure. I think blinatumomab is fantastic and revolutionary, but not fully so if it can't reach every patient in need. And I think this is something that we as a community have to take on as an effort and a priority because it does need to reach every child regardless. I mean, I feel like we talk about all the prognostic features. The greatest one is where you're born, and that needs to change in terms of access and opportunity. We have patients here who live in very rural settings. Even if I can write you to get blinatumomab, but you can't literally do it because you live 8 hours from the nearest metropolitan center. And so, I think it's going to take involvement of pharmaceutical companies, governmental agencies, and pediatric oncologists around the world to change that state of affairs. But I don't have great answers right now, but I think those are answers that we have to come up with. So thank you.
Right. Excellent.
Good afternoon, everyone. Also, thank you to the organizers for the opportunity to speak today, and thank you to Wade Kiono as well, who we've collaborated with a few patients on for cell therapy as well over the years. So I have nothing to disclose. And for the purpose of my talk today, probably not too surprisingly, I'm going to be focusing mostly on chimeric antigen receptor T cells, or CAR T cells. And I think for this audience, many are aware, but just as a quick reminder, so these are patient-derived T cells collected from patients that are then engineered, usually through lentiviral transduction, to express a chimeric antigen receptor, or a CAR. That was showing up. Oh, shoot. Sorry, guys. With the external portion containing an scFv linker, which can be designed to target the tumor antigen of your choice, and then that's linked to a intracellular co-stimulatory domain as well as a T cell activating domain, which then turns the T cell on, makes it cytotoxic, and then can kill the tumor cell. And so I remember being a fellow in the early 2010s when this first came on the horizon, and everyone was so excited about this, and the first few patients treated with CAR T-cell therapy showing remission induction after being refractory to multiple lines of chemotherapy or bone marrow transplant. And sort of after this discovery came just a little explosion of phase 1 trials looking at CD19 CARs in pediatric B-ALL, all of which showed pretty tremendous remission induction rates, and was pretty remarkable, again, in this highly refractory group of patients. And that led to the global phase 2 registration trial of tisagenlecleucel, now Kymriah, using the product derived out of PEN or CHOP, of CD19 CARs in B-ALL, and then that ultimately led to the FDA approval of the first gene therapy, which is remarkable that it's in pediatrics, using Kymriah for B-ALL, and that was in August of 2017. So what have we learned since then? It's been 7 years since that happened, and so for today, I'm going to try to cover, and there's a lot, so I'm going to try to highlight the updates that we've learned since then and how effective really are CAR T-cells in B-ALL since that approval. What have we learned about patients who have risk factors of resistance to CAR T-cells? What have been the next generation of cell products for B-ALL, currently in investigation, and then what are some future ideas? And so to do this, I'm going to be reviewing data from the PLAT series of trials that were out of Seattle Children's, also the tisagenlecleucel trial data from CHOP and Novartis, data from the Pediatric Real-World CAR Consortia, which is retrospective look at commercial Kymriah use, and then a consortia called the Carnation Consortia, which also looks at retrospective data in pediatrics. It's really a mix of investigational and commercial product data. And so first, I think looking at a couple of the long-term results. So the Eliana trial was that phase 2 global registration trial of tisagenlecleucel. And what we've learned is that even though these products are highly effective at getting patients into remission, which is very crucial, at about a year, 2 years, 3 years out, about half of patients are experiencing a disease event or a relapse. So the duration of remission is about 50%. And this is shown across trials. This was our PLAT-02 trial, which was investigation of another CD19 CAR, very similar to Kymriah. And this was our phase 2 data that's not yet published, soon to be published, showing pretty similar 2-year event-free survival, leukemia-free survival rates. Perhaps numbers are a little bit higher than the Eliana trial, but I don't think this is necessarily because the CAR is better. I think that we've evolved in learning how to treat patients with lower disease burden and also getting better at figuring out who needs to go on to, for example, a consolidative bone marrow transplant. And so why do patients fail? And I think there's 2 big buckets we can put patients in as to how they fail CD19 CARs. There's a small proportion, probably around 5 to 10%, that just don't respond up front. So we call them dysfunctional or non-responders. So they either experience no CR at all, or they have a fleeting CR and then have recurrence while CAR T-cells could even still be present. So a smaller group of patients. And then we have, of course, our post-CAR relapse patients. And these post-CAR relapses can come in a few different flavors, if you will. So we have patients that still express the tumor antigen that are CD19 positive. And those patients are probably failing mostly because the CAR T-cells are no longer persistent in vivo, which can be for a few reasons, perhaps immune rejection or exhaustion of the T-cells. And then, of course, we have our antigen-negative patients, so where the leukemia no longer expresses the target of the CAR. And so on the right are sort of kind of concepts about how you might envision getting around those challenges. And then a subset of CD19 negative relapse or lineage switch, where patients will subsequently have target negative relapse that can also evolve from being an ALL phenotype to an AML phenotype. And so I want to try to walk through kind of what have we learned about some factors or predictors, and I'm going to start with the non-responders, so that smaller group of patients. So probably there's some product intrinsic factors, so T-cell fitness, in other words, and then also leukemia intrinsic factors as to why some patients don't respond to CARs. I'm not going to go through all these in detail, but just briefly, Finney et al. Identified a T-cell phenotype in patients of the collected apheresis product that were shown to have high expression of a marker called LAG3, low expression of TNF-alpha that was predicted to have patients that did not respond to CARs. And then other groups have shown, for example, there's a subset of patients, and this is samples from Plateau 2, that had a signature that was associated, even though they had a BALL phenotype, their signature was more stem or myeloid cell-like, and these patients were also predicted to not respond to CARs. There's been work showing that some tumor cells, some leukemia cells, have impaired death receptor signaling, which might be critical for CAR function, and so those patients are also enriched in non-responders. And then another factor which I think has come up, and I'll talk a little bit more about, is this risk of non-response to blina. And so as Dr. Rao mentioned, blinatumomab is being increasingly used in patients, especially in the upfront setting. And so there's data to show that if patients are non-responders to blinatumomab, they are more likely to be non-responders to CD19 CAR. I will emphasize, though, that's not a causation. There's no data to show that non-response to blina causes a non-response to CAR. And I think rather it's more likely indicative of a immunotherapy resistant signature. And it's important to note out that in this graph on the right, in the blue, patients that didn't respond to blina still had a 60% chance of responding to CAR, so still worth a shot to give that CAR a try. In terms of other disease factors that could impact response, or now thinking about risk of relapse, one thing that hasn't panned out, which is really interesting and has been shown across groups, is our traditional high-risk cytogenetic groups. So things that we think impact chemotherapy resistance have not necessarily panned out to show immunotherapy resistance. So high-risk groups like hypodiploid, pH-positive, pH-like, et cetera, have had similar complete response rates to CD19 CAR, as well as no difference in relapse-free survival. So other disease factors, so one thing that has panned out, conversely, across groups, is that we've learned that high disease burden prior to CAR T-cell infusion is associated with inferior outcomes, and the most commonly used cutoff is 5% blasts in the marrow before CAR T-cells. And so the top row is data from our Plateau 2 efficacy cohort showing that in the blue, these are patients with low disease burden compared to red, which is high disease burden, and across the board, event-free, leukemia-free survival, and overall survival were inferior with high disease burden. The middle row is the retrospective data from the Carnation Consortium showing the same thing. And then the bottom row is from the Pediatric Real World Consortia looking at commercial Kymriah. Again, similar trends across the board. I think what we don't know is high-risk disease burden a modifiable risk factor, or is it rather a biomarker of really high-risk disease? As people know, if you've sent patients or have patients that have gotten treated with CARs, you enroll them or you send them their product if it's a commercial product, and then you have a few weeks to bridge until they actually get their CAR infusion. And we get asked this question a lot: can we just give them really intensive bridging so we can knock down their disease burden? And I think that we still have data yet to understand. We need to create data to understand if that lowering of disease burden actually makes a difference and can actually put a person in a different category where they would have an improved outcome with CARs. The other thing that's really interesting is looking at the category of patients that get these CAR T-cell products. We have relapsed or primary refractory patients. And so the Carnation Consortia showed that if you were primary refractory in red, you actually had an improved survival post-CAR compared to those that were relapsed patients. The Pediatric Real World CAR Consortium did not show this. They actually showed no difference in relapse versus refractory patients. However, the refractory cohort was more enriched for patients with higher disease burden, so it was a little confounded. And then in Seattle, our Plateau 2 results from the phase 2 group actually show small numbers, but the blue line is the refractory patients, so primary refractory versus red and relapse patients. And I think it's just worth pointing out this curve. I think it's really remarkable to think about the primary refractory patients up front and what their LFS or EFS could be with CAR. And what I don't have a slide on is showing COG trial AALL 1721, which is looking at the incorporation of Kymriah in a select group of high-risk patients who have including those with residual disease at the end of 2 courses of chemo, which would fall sort of into this group. And data from that trial are clearly pending, but I think it'll be interesting to see if it pans out similarly. And so what about after you've gotten your CARs? Are there other risk factors that predict inferior outcomes? And so what's been nicely shown, again, across groups, is that if you have early loss of functional CAR T-cell persistence, which is typically measured by the loss of B-cell aplasia, so as a surrogate marker for functional CAR persistence, if you lose that persistence, you have an increased risk of CD19 positive relapse, which makes sense. And then the Eliana trial was shown that the earlier you lose it, the more inferior your survival is likely to be, which also has been shown across groups. So the question is, what impacts the duration of CAR T-cell persistence or B-cell aplasia? And so a few things that have panned out. One is if you get proper lymphodepletion with flu-cy, that probably matters, and it's been shown to have improvement in CAR persistence. So much so that actually the dosing and the exposure of fludarabine also matters. And so coming soon is a phase 3 fludarabine optimization study based on prior data called the Influence study that's going to be led by Kevin Coran out of MSK to look at optimizing the flu dose. And then the other things that we and others have shown is that if you go into your CAR T-cell infusion with low antigen burden, so the sum of your malignant and non-malignant CD19 cells is less than 15%, you have inferior CAR persistence, so could lead to increased risk of relapse. And then the last thing that we showed in our Plateau-2 data is that if you have this rapid early contraction of CARs soon after the CAR infusion in peripheral blood, you also are more at risk to lose your CAR T-cell persistence sooner, which makes sense if you think about it. And so these are some of the risk factors we've thought about. How could we overcome these? And so one approach we took in Seattle is this sort of pilot study of a novel cellular product called T antigen presenting cells, or TAPCs. And this is basically going back to the patient's T-cell product stored in the freezer and just having it express a dummy CD19, a truncated CD19 protein, essentially to act as like feeder cells in vivo following CAR T-cell infusion. And the hope was this might be able to overcome low antigen burden and promote ongoing persistence. And this was the design of the trial. Patients were in one of two cohorts on the trial, either the risk factor of low antigen burden or those rapid early contractors. And patients could get these big bolus doses of TAPCs, so really high antigen exposure in these episodic fashion, roughly every 4 weeks for up to 6 doses. So what did we see? This graph is a single patient's experience. So the blue is detection of the CAR T cells. The green is transient detection of the TAPCs, and they're gobbled up quickly by the CAR T cells if they're still around and functioning as they should be. And what was interesting is if you look after some of these TAPC infusions, which are the red triangles, we actually did see a secondary expansion or reactivation of the CARs in vivo detected by flow. And then did this translate to anything? So it sort of did. It was, again, low numbers, but we did see a trend towards improved persistence in patients, both the low antigen burden patients and the rapid early contractors if they got TAPCs. And data is still, it was a pilot study, but so far encouraging. So switching gears just for a minute, I mentioned antigen escape, and so the thought there was that maybe 2 targets are better than 1, and knowing that most B-ALL expresses CD22, there was a little explosion of trials all over the world, really, that took place looking at targeting both CD19 and CD22 simultaneously. There's a couple different ways you can do this, which I've shown here. You can have co-administration of 2 different products, 19 and 22 CARs. You can have a single T cell that expresses both CARs. You can do co-transduction, so you have a mixed population. Some cells only express 1 CAR, some express 2. And then you can have this tandem idea where you have 1 single intracellular signaling domain, but 2 SCFVs on the surface. And so in Seattle, we chose the co-transduction approach. This was our Plateau-5 trial. And so we actually had, long story short, 2 versions of this CAR. The short story is that version 1 had a dud 22 CAR, so we moved on and modified that and had a version 2 of the product. But what was really interesting is that, first and foremost, we saw a pretty similar MRD negative CR rate with this product compared to CD19 CARs of 87%. But we actually had patients who had dual antigen escape following this CAR. And then what was sort of an interesting observation is that in version 2, the green bar is the 22 CAR in vivo over time in patients. And it really expanded out of proportion to the other CARs. And why this happened, I don't think we know, but the 19 CAR was kind of lost in this model. And then there are other ongoing CD19 by CD22 dual targeting CARs at Stanford. They've also seen excellent tolerability and good response rates with their 19 by 22 CAR, and I think they have a couple slots remaining in their trial, and that's run by Leora Schultz. And then there's an upcoming trial looking at patients who meet the label indication for Kymriah, followed by preemptive 22 CAR a month later. That's going to be run by Kara Davis, and that's the sequence trial. At CHOP, led by Regina Myers, there's a phase 1/2 combinatorial humanized CD19 CAR plus 22 CAR, so giving those two products at the same time. And they're enrolling both CAR-exposed and CAR-naive patients, currently just CAR-exposed. There's an ongoing bispecific CAR at NIH with Nirali Shah, and then Colorado just opened a newly opened bispecific 19 by 22. I'm sure I'm missing some, and then a lot of ongoing work yet to hopefully be data to be coming out soon. And then just briefly, I think as we're getting along just with all these risk factors, it's going to be crucial to understand which patients are going to benefit, which patients really need some consolidative therapy, for example, a transplant. And so just in terms of after CAR, what can predict frank relapse? I think it will surprise probably no one that having any detectable MRD after CARs is predictive of impending relapse or frank relapse, but what time point? And so it was shown that at the 3-month time point after CARs, if you have any detectable MRD by the most sensitive method possible, that was incredibly predictive of ongoing, future frank relapse. And so what to do about this, I think Marie Bleakley will talk about next after me, this upcoming CAR cure study to investigate how to manage this. And so lastly, just one thing to mention, some patients can't go on and get consolidated transplant, but it's clear they might benefit. They're at risk for relapse. And so what else could they do? And so I think anecdotally, a lot of us have been doing things like maintenance chemotherapy after CARs in patients who have early loss of B-cell aplasia. And so there was a report published just this year looking at this retrospectively and looking at patients who got either transplant or maintenance chemotherapy after CAR, and actually showed in this small numbers, but still no difference in EFS and OS. And based on this and the fact that I think people are just doing this anecdotally for some patients, there's a plan for a prospective study called Endurance, and it should be done in collaboration with US and European sites looking exactly at this. So if patients have early loss of CAR T-cell persistence or B-cell aplasia after CARs, they would be eligible to enroll in the study and get maintenance therapy. So in conclusion, I think one thing to note, since FDA approval of Kymriah, leukemia events we've seen continue to occur, and they can occur late following this first iteration of CD19 CARs. We're continuing to work on identifying product, disease, and patient intrinsic factors that impact outcomes following CARs and BALL, and this work is still very much ongoing. I'm sure I missed many reports in all of this. And just to take a minute, I think with increasing blinatumomab use up front in BALL, again, it's important to note that association of non-responders with increased risk of inferior outcomes with CAR, but it's not necessarily associated with antigen loss. And I think really is indicative more, like I mentioned, of this immunotherapy resistant signature. I think with increasing blina use up front, less patients overall will probably relapse, which is great. But I think we should be prepared to see an increased number of extramedullary relapses, and CAR could be a good fit for that. I think there's still very much a role for 2 targets being better than 1, but I'm not sure if we've demonstrated so far that 22 is the best second target to improve outcomes over 19 CAR alone. And so we need ongoing work in alternative novel cell therapies for BALL. And then I'm just going to throw this out there because I think we've been thinking about this a lot lately. With the current CARs that we have, I guess my question is, why do we have such a high bar for CD19 CARs to be a standalone therapy for everybody? I think we were really fortunate that some people have durable cures after CD19 CARs, but we don't have the same expectation of other immunotherapies. And so just a challenge us to think about with the current CAR therapy that we have, until we have a better one that comes along, thinking about multimodality therapy for these patients as well, and some risk-adapted approach. And that might actually be a way to think about long-term cure for relapse refractory patients, and maybe even a way to move this closer to upfront therapy if possible. For example, could you imagine a little debulking chemotherapy regimen followed by CAR, close NGS monitoring, and even maintenance therapy for these patients? So with that, I have many people to thank, and then I'll take any questions, but I think I ran a little over, so.
Again, we'll take one question, and then we'll try to have some time at the end.
Hello, thank you for your talk. I know this is a high-risk B-ALL talk, but I'm wondering if you can comment on how far we've come with developing a CAR for T-ALL. I know there's been some emerging data on using allogeneic CAR T-cell therapy. Can you comment on that?
Yeah, so for the first part of your question, didn't have data on T-ALL for the purposes of this talk. It was out of scope for this talk, but thanks for the question. And I will say in Seattle, we have not had ourselves some T-ALL trials. However, I know especially with Baylor has really helped promote this work. And I think that it's pretty exciting, especially their CD7 CAR for T-ALL has shown some promise, and I think trials are ongoing, but I think there's definitely more excitement there than maybe there was initially with some of the other targets. So hopeful to see where that lands. And then I think for the allo question, it's a really good question. There was a little burst of activity for a while about, getting off-the-shelf CARs for BLL. And I think the problem there is thinking through persistence. And maybe depending on how you want to use CARs in the future, persistence isn't the necessary equation based on that last thing I just mentioned. But if we're aiming for durable persistence, allos haven't been shown to have that. So I think until we can, and there are people that are working on modalities to make those CARs more fit to be more durable in a variety of ways. But I think until we can figure that part out, there's still nothing to replace the autologous CARs.
Good afternoon. I really appreciate the opportunity to participate in this wonderful meeting, and I'd like to thank Dr. Kiona for the invitation for our group. As the last speaker of the day and co-chair, I'm planning to keep this very short and leave about 10 minutes for questions for the whole panel. I have no disclosures relevant to this talk. I'm going to start by sharing some encouraging data from the Center for International Blood and Marrow Transplant Registry, which shows quite favorable outcomes of transplantation for most pediatric ALL patients in the current era. Specifically, among approximately 1,700 patients who underwent transplantation for ALL between 2016 and 21, but 3-year overall survival for patients transplanted in CR1 was approximately 80%. For CR2 and beyond, it was 72%, and for patients not quite in remission, it was a little lower. So those data are really quite encouraging and an improvement on previous eras, but there are important questions remaining. For the sake of time, I'm going to focus on just 2 questions that come up quite a lot in our high-risk leukemia program and discuss the data around them. So the first question relates to the outcomes of alternative donor transplantation, and the second is about the use of transplantation after CAR T-cell therapy. So in the first scenario, we're presented with a patient who has an indication for transplantation, for example, a PH like ALL with measurable residual disease at the end of consolidation. But they don't have an HLA-matched sibling or 10 out of 10 unrelated donor. And we're asked to comment on whether transplantation with an alternative donor is associated with a lower probability of success. So my first comment is this isn't unusual these days. So in the US, approximately half of our patients receive transplantation from alternative donors. Specifically in the CIBMTR data set, 25% of patients under 18 received haploidentical transplantation, 14% umbilical cord blood, and 3% mismatched unrelated donors. interestingly, as shown in the graph on the left-hand side, we can see that the use of haploidentical transplant in bright blue has really dramatically increased over the last decade or so. And as shown on the right, this is true for ALL as well as AML. But what are the outcomes of alternative donor transplantation for pediatric ALL? So we can see on this slide that the outcomes of cord blood transplantation of ALL are excellent in the current era, at least in large institutions that regularly perform this type of transplant. And here I'm showing the outcomes of 25 consecutive pediatric AYA patients with ALL transplanted at our centre in the relatively recent past. This is a mixed population of different remission stages, and a good number of them also went to transplant with measurable residual disease. With a standard fludarabine, cyclophosphamide, total body irradiation conditioning, and a cord blood unit, mostly just a single cord blood, and post-transplant immunosuppression with MMF and cyclosporin, you can see that the leukemia-free survival was 87%, and there was actually no relapse in that cohort. I have Anne Dahlberg and Ted Gooley from our centre to thank for allowing me to show this unpublished data. The current outcomes of haploidentical transplant are also encouraging. In this multi-centre Paediatric Transplantation and Cell Therapy Consortium prospective trial that was published in Blood Advances last year, there were 11 patients with ALL included, and they received myeloablative TBI and cyclophosphamide twice before and on days 3 and 4 after transplant, the latter for prophylaxis of GVHD, and also received a graft from a haploidentical donor, tacrolimus and MMF. The leukemia-free survival for the ALL patients was approximately 80%, and relapse was lower actually for the ALL patients than the AML. And although I'm showing the data for the haplotransplant with PT-Cy, there are also very encouraging published data for selective ex vivo T-cell depletion with alpha-beta TCR and B-cell depletion and naive T-cell depletion. To summarise the information relevant to question 1, the outcomes of alternative donor transplantation for ALL are very favourable in the current era, and there's no need to defer transplantation for ALL patients that don't have an HLA-matched donor. And both cord blood transplantation and haplo are very reasonable options. Of note, importantly, there's a prospective randomised trial comparing haplotransplant either PT-Cy or alpha-beta T-cell depleted with HLA-matched unrelated donor transplant that's being performed by COG, and that's ongoing. Now, the second question, a patient has CD19 CAR T-cell therapy for refractory B-ALL and is now in complete remission. Do they really need a transplant as well? So I'll first point to this data by Corinne Summers at Seattle Children's that shows that consolidation therapy with transplantation after CD19 CAR T-cell therapy is associated with a survival advantage, at least in some patient groups. Specifically in this single-centre study of patients that received a 4-1BB CD19 CAR T between 2014 and 17, in which 91% of the patients achieved an MRD negative CR. There were 64 patients enrolled, 50 were alive in an MRD negative remission at 63 days. And then the leukemia-free survival was significantly better for those that received a transplant compared to those that didn't. Those that had not had a previous transplant before the CAR T-cell therapy also had a significantly better leukemia-free survival with a transplant rather than without. But those that had previous transplant, there was no significant difference. But if they lost B-cell aplasia early, they had an inferior outcome. So these data inform our current practice of offering transplantation at 9 weeks after CAR T cells for patients that have not had previous transplantation. And for those who have had previous transplantation but lose B-cell aplasia early or become NGS positive. And similarly, in these data that were compiled from multiple centres and presented at ASH last year, on the question of transplantation after CD19 CAR therapy, it's very clear that, as shown in the bottom of the table, that patients that receive a CD19 CAR with a CD28 co-stimulatory domain will almost all relapse without transplantation eventually. And there's also, in the top half of the slide, there's fairly clear, consistent trend across the data sets that with 4-1BB-based CAR T-cell recipients, those patients that don't receive transplant, those patients who do receive transplant are less likely to relapse than those who don't. So those data together support the current practice of recommending transplantation for many patients who have received CAR T-cell therapy and have not had a prior history. But as we're all aware, the late effects of transplantation can be very detrimental to children and sometimes devastating. So there's been interest in developing an alternative approach involving very close monitoring of recipients of CD19 CAR T-cell therapy, monitoring them for loss of B-cell aplasia, which, as Colleen said, is a proxy for loss of functional CAR T-cell persistence, and for the presence of very low-level residual disease determined by next-generation sequencing MRD testing of blood and marrow. And then using transplantation only preemptively as required based on those monitoring. So those thoughts are informed by the data that was led by Mike Pulsifer and published in Blood Cancer Discovery, the top graph showing the association between loss of B-cell aplasia. The sooner you lose it, the lower the chances of leukemia-free survival. And the bottom graph, which shows the presence of NGS MRD at almost any level or at any level, inevitably leads to relapse. So in order to prospectively evaluate the question of whether NGS and B-cell aplasia could be used to guide a preemptive transplant strategy for 4-1BB CD19 CAR T-cell recipients without increasing the rate of morphological or flow cytometry relapse compared to historical direct-to-transplant recommendations, we developed a clinical trial called CARCure, which is currently enrolling at 6 or 7 centres across the US. And patients are enrolled within 6 weeks of receiving a CD19 4-1BB CAR T if they have, at that time, B-cell aplasia, no evidence of residual disease, and a BALL clone that can be tracked using NGS. They're monitored very closely with blood tests every 2 weeks and marrows at regular intervals. And if they lose their B-cell aplasia or become NGS positive, they're recommended to receive transplantation. And the aim is to see if we can transplant less children, but to collect the data very closely and figure out the risks and benefits of this sort of preemptive strategy. So in summary, transplant outcomes for ALL in the current era are generally very favourable. Suitable donors can be found for almost all patients, and alternative donor options have generally very encouraging results, although the randomised trial of haplo versus matched unrelated donor transplant are pending. Most of the data suggests that recipients of CD19 CAR T-cell therapy have a better probability of durable remission if they receive consolidative transplantation. But those recommendations may be refined pending the outcome of prospective trials evaluating intensive monitoring of CD19 CAR T-cell recipients with NGS MRD testing and B-cell aplasia evaluation. So thank you for your attention. Thank you very much to my colleagues in the high-risk leukemia and BMT programs at Seattle Children's Fred Hutch, to the Siop organisers, and in particular Dr. Kiono, and very much to the patients and the families who trust us to take care of them and evaluate ongoing improvements for their care. I'll move back and we'll take questions for the whole group.
Why don't we take one or two in person, and then we've gotten a number of virtual questions, and perhaps we can take one or two from that. Any questions?
Thank you. Richard Mitchell, Sydney, Australia. Thank you very much. I think really fascinating look at the high end of high-risk BALL. I kind of want to follow up on my question from my colleague from India before in a slightly different manner. I run the unit in Sydney, Australia. I'm a BNT and CAR-T doc down there. We, even in a high-income country, have struggled to deliver some of the therapies you guys are talking about, even though we have access to them all. We wanted to participate in CAR-Cure, and Maria and I nearly got it up and running, and the barrier was resourcing to even get the testing done. In Australia, Novartis opened a production facility and then closed it 12 months later because they were not able to make enough money. So I'm interested in all of the panel's opinions about, as we move forward with these new and exciting therapies, how do we make them sustainable? Obviously, we've already heard that from a lower middle income country point of view, that's a challenge. But we're having trouble as well. And we know that in many, particularly for cars, for example, it's a real challenge to make sure that these products are, are made sustainably because it's such individual medicine. So I'm interested in everybody's opinion about, as we move forward, yeah, we can do it and you can do it on an individual basis, but how do we make it sustainable? How do we make sure these therapies become standard of care that we can deliver long term and then hopefully make them accessible to all of our colleagues all around the world?
Is this on? Yeah, okay. No, thank you for the question. I mean, I think it's such an important question. And similar to Dr. Rao, I think it's hard not having all the answers now, but recognize it's a problem. I think, there's a couple of endeavors underway now, at least I'm thinking about CARs in the moment, in terms of thinking about, so access is multifactorial. It's financial, it's proximity to centers that can treat your patients, it's the ability to actually collect a product sometimes from really sick kids. And so I think all of these are being addressed in different ways to some degree. I think like off-the-shelf product questions that were asked is one potential modality that could, if refined, help with some of that. I think that the kind of decentralized manufacturing concepts that are taking off with the Miltenyi, but these are still not perfect answers. So I think it's going to take, some time, and again, like Dr. Rao said, kind of collaborative efforts from the pediatric oncology community, pharmaceutical companies, the FDA, regulatory authorities to make products that are more available and more sustainable. But I wish I had a better answer for the time being.
Hi, Chi Kong Lee from Hong Kong. I would like to ask about the post-CAR-T consolidated transplant now. It appears to improve outcome, but will be associated with more late effects. So would the conditioning for the transplant be adopting, say, non-TBI or non-myeloablative approach to improve the outcome?
So right now, the general recommendations are myeloablative TBI, at least in patients 4 and above, based on the Forum study and randomized control trial that studied hundreds of patients across the world and found a 17% survival advantage to myeloablative TBI compared to chemotherapy only. However, that's not the answer that the community wanted. We would like to avoid myeloablative TBI in growing children and in patients in general. And there's a concerted effort going into studying the potential to transplant patients in the deepest remissions, so in next generation sequencing MRD negative patients with either no TBI or lower dose TBI. Mike Pulsifer and colleagues have published some relatively recent data with alpha-beta T-cell depleted transplants with reduced intensity conditioning that look pretty good in single-arm small studies. But there is an ongoing NRAD study that's looking at this more rigorously, and we're looking forward to that result. So right now, the recommendation is yes, myeloablative TBI in most circumstances.
I just wanted to go back to the first question, if that's okay. I'm not going to sort of talk about cars. I've had discussions with different companies, and we've had struggles with trying to get an agenda forward for certain products, and that's just a whole problem. I don't know. I think the future is bright and sustainable for blinatumomab. As you probably know, Amgen is also conducting studies where they're pushing the dose and they're giving it sub-Q. The trial that we ran, which we can't really talk about, it's a large trial, and we had to deal with a lot of pump issues, a lot of other things, and Rachel may deal with some of the questions that are coming there. But the other thing that I talked about a little bit, and I get the fact that in resource-limited countries, it's hard to get that drug. We do pretty well with chemo, right? I mean, we do pretty well with standard chemotherapy, and the overall survivals are also really good. So maybe you prioritize blina for patients who relapse if you just can't provide it for every single patient who's diagnosed in your country. And maybe that's an unsatisfying answer, but maybe that is the answer. So I don't have all the answers, but maybe that's something to consider as well.
I wish I was keeping track of who was first. But...
I think she was before me, before him.
Okay.
Thanks, yeah.
So go.
Thank you. Thank you for wonderful talks. My question is more directed to Dr. Rao. When we're talking about more single agent, my name's Ingrid Liljenthal from Stockholm, and I was just wondering, when we're talking about more single agent upfront therapies, what are risks for resistance, and have you seen any resistance in like the blinatumomab studies to the single drug?
Right. So I would say most of the trials use blinatumomab in combination with at least some induction therapy, because one thing that we have learned about blina is it's most effective in a lower disease state. Certainly if you have above 50% blast, the response rate is going to be much, much lower. So at the very least, I would say it's used in combination with something that would get the disease burden down before you would get blina. But I think there are data to suggest that you don't need tons and tons and tons of other chemo, so I think that's an evolving question. As far as resistance to blinatumomab, I think that's going to be super important. I think we're going to need to redefine how we prognosticate for patients, because it's not yet clear if the same genetic factors that Dr. Lowe talked about that are predictive of relapse with standard chemotherapy alone are going to hold for patients now treated with blinatumomab. Dr. Annesley talked about data showing that those same genetic factors are not predictive of response to CAR T, so I'm not sure how those are going to hold up, but we need to answer that question with data. There are some patients who we know from the 1331 trial maybe didn't have a sustained response to blinatumomab, so perhaps that's a T-cell exhaustion issue, and so studying the PD-1 inhibitors to maybe enhance what the T-cells are able to do is another approach that's being looked at as well in our currently running upfront relapse trial. Right now, we haven't defined, I think, the predictors of blinatumomab resistance, but it's going to be something that we have to do now that it's being incorporated into upfront therapy more and more.
Sorry, Siobhan Cross from New Zealand. I'm really interested more in the genetics and molecular side of things. Whole genome sequencing is again outside of the realms of many places, and that's not necessarily money. They're getting cheaper all the time, but the amount of bioinformatics and the personnel and setting that up is just unachievable. Is there any work going on to try and pull the important things of what we do know into panels that could be more usable? Because global health is a continuum from have no treatment options to what we've been talking about today, and having some panels to help improve this risk stratification would help with that.
I think that's a great point as well, and I think that technology, we're always trying to absorb advancing technology into standard clinical practice, right? That's the challenge I think that people who are really interested in these types of assays face all the time. And so I remember when Steve was the chair of ALL and we were working on how to identify pH-like and how it was so involved. It's still involved. But, we try to combine sort of standard fish assays. Can we get as much information as we can from a fish assay, which is relatively standard, et cetera? And the other thing that's sort of come down in price as we watch, yes, whole genome sequencing is very labor intensive, and there are very few places who can do it well and quickly. RNA transcriptome sequencing has actually come down quite a bit in price, and so we're looking into ways, and Dr. Rao can speak more about that, into whether we can also look at patients through a combination of, sort of standard assays, but also transcriptome sequencing. So I think there are creative ways. Sure, in the ideal world, it would be great if we could do a whole genome sequence on everybody, and that's, I don't know whether you're still thinking about that, but no, I understand the complexities there as well. But so we try to combine different assays where we can look at the strengths and the sensitivity and specificity, et cetera, and sort of put that into algorithms that work, knowing that we will continue to evolve. Every few years, we got to take a look and see what else is coming down the pipeline. If you want to add something.
Yeah, I don't have much to add. And I think, we're looking at the possibility of adding RNA-seq to all of our newly diagnosed patients, as is being already done in some consortia around the world. But I still think we're going to have to identify the precise lesions that we're going to use for risk stratification, while in the background we're going to be able to collect a lot of data. So I think there's sort of the question of what you do with patients enrolling on a clinical trial, and then the questions of how you appropriately risk stratify your patients on a day-to-day basis. And I think that can be done with assays that are specifically designed to detect the lesions that are useful for prognostication at the time you're treating patients. So I think multimodality is where we are right now with that.
Hi, Rob Peters from Princess Maxima Center in Utrecht. In our team, there's some hesitance to do CAR T cells if patients are MRD negative, because you need a certain target level to be active. Is that correct, or should we simply use the CAR T cells if patients are MRD negative, real MRD negative?
Yeah, it's a great question. I think it's a hard question to answer. So I think in the first part is that I think that the fear is that could lead to like an inferior persistence because you don't have antigen to drive persistence. What has been interesting about some of the patients we've enrolled, at least in our trials out of Seattle, is that there's a fair number of patients, depending on when you collect them and depending on when you treat them, really, rather, I should say, is that they still have a decent amount of CD19 antigen, which I think that is the true predictor. So I think it's a different patient that's MRD negative that has CD19 positive B cells around to drive some expansion and persistence versus a patient who is B cell deplete MRD negative. Those are the ones that I worry about. And I don't know that so I think, and that we've had patients that could benefit from CARs in that situation. For example, they have CNS disease, and that's still something that can be achieved as eradication of CNS disease in that situation. But I would worry about that patient having longer-term persistence, and that would be somebody I would follow very closely and consider one of those post-CAR consolidative approaches for right now.
Luis Argüelles from Mexico. Going back to the very first question in this session, for those interested in learning how to make these sophisticated treatments available for people living in low and middle income countries such as CAR T cell production and bone marrow transplantation, I suggest to remain in this room, because after the break we will address those critical points which will benefit a lot of people all over the world. Thank you.
Thank you.
Thank you.
Great. Thank you, everybody. Really appreciate your time. I hope you found it helpful.